The scientific method. As graduate students, it's imperative that you begin using the scientific method to analyze research articles for efficacy of dietary supplements and ergogenic aids. So let's run through the scientific method. There are 5 components here that we're going to look at individually. So the first one being identifying the research question. So what is the problem that you're trying to look at? So you identify the problem, then you're going to search the literature review, and then you'll identify variables that you're interested in. So for example, a variable is going to be a dietary supplement, and then another variable might be the outcome. So if you wanted to look at branched chain amino acids and muscle mass, those would be your variables. And we'll talk a little bit more about that in another lecture called Research Terminology. So you identify those variables you're interested in. And then based on all of this information, what you're going to do is state your hypothesis. A hypothesis is basically a yes or no statement. That is, you make a statement, yes, this supplement will work, or no, this supplement will not work. So it's very simple and straightforward, and something that will be the basis for identifying what the research project is all about and whether or not you're going to use the data collected to support or refute that hypothesis that you developed. So the second part of this is going to be to design the study. So you need to come up with your study protocol. What is it that you're going to look at? How are these variables that you're interested in? How are you going to set them up to actually test for changes in the variables? So you'll also need to choose a sample population. Are you going to look at athletes? Are you going to look at a clinical population? What's the population that you're going to look at? The third aspect of the scientific method is to look at methodology. So we need to collect data. How are you going to collect that data? Are you going to use surveys? Are you going to use blood samples? Are you going to use urine samples, etc.? You'll need to compile that data that you collect into tables and perhaps forms if it's a survey study. So you need to think about all these aspects and then put it all together in one neat package so that you can actually do the next part of it, which is the data analysis. So you need to be able to analyze all those data that you collected and then interpret the findings. What do the findings mean? Is there anything there? How do you do that? And from those interpretations, can you draw valid conclusions? So going back to the hypothesis, which is typically represented in statistics as H sub naught, so HO here, and H1. So HO is your hypothesis, so you've made this statement, and you are either going to, based on your data, reject HO, so you either reject your hypothesis or you fail to reject your hypothesis. And those are the terms that are typically used. You really want to avoid using the word prove or proof. That's kind of a banned word in the scientific community because it implies that something is in fact a fact, which there are very few things in the scientific world that are actually facts. For example, the fact that the Earth revolves around the sun in the solar system. That's a scientific fact. So we want to be careful and distinguish our statements from scientific fact versus scientific theories, which aren't necessarily proven. So we need to be careful with that. So we're either going to reject our hypothesis or we're going to fail to reject it. Now, if we reject our HO or our hypothesis, then what we do is we accept the alternative hypothesis, or H1, which is basically that it's the opposite of what your original hypothesis was. So if your original hypothesis was that amino acids increase muscle mass, but you did not find that in your study, so you reject that hypothesis, then you'll accept the null hypothesis, or H1, which is basically that amino acids do not increase muscle mass. The last step which is very, very important, is dissemination of information. You need to share your findings with the scientific community so that we can contribute to our understanding of this particular area. If you do a study and no one knows about it, then basically that information is not going to help others sort of think about this information a little bit differently and perhaps research it further. We also should suggest further research. Oftentimes when we do a study, you end up coming up with more questions than answers. So it's important to suggest, well, we should look at this aspect of this particular project because we didn't do that, and that's important. We also want to publish our work in respected journals, as articles, as abstracts, give oral presentations, give poster presentations at scientific meetings. This is very, very important, again, because of the dissemination of information so that we can come together as a scientific community to figure things out. For students, usually this comes in the form of a thesis or a dissertation that then you go ahead and turn into a manuscript and get it published in a journal, or perhaps you present it at a conference, whether it be in print form or orally. So this is all an important process, part of the scientific method. And a lot of times what you'll find with these dietary supplements is that you won't be able to access the data that the manufacturer of that particular substance claims that they've collected. You won't be able to see that article. So you won't be able to make a judgment for how the study was conducted, whether it was conducted properly, and what the findings actually mean. So this is an important part of the process. Now the last slide here talks about the assumptions of the scientific method. Nothing is perfect. The scientific method is good, but there are still some flaws with it. And it assumes that nature is going to be orderly and regular. In other words, events that you find when analyzing and collecting data are going to be somewhat consistent and predictable, that you can replicate the study results. Oftentimes, you'll see individuals use one particular article as the basis for the argument for the use of, let's say, a dietary supplement. However, if you can't replicate those results regularly with future research projects, then it was probably an anomaly, or the study was designed improperly. So the scientific method assumes that you're going to get consistent and predictable results. Of course, this isn't always going to be the case. The second assumption here is that events or conditions are not random or accidental, that by manipulating one variable, you can actually affect other variables that you're measuring. So one or more causes can be discovered. It's supposed to be grounded in reality and not personal bias or a subjective belief. But again, as we'll talk about in another lecture, based on evaluating the scientific literature here, we'll see that sometimes authors or scientists can be biased and introduce that bias into their research project, which presents a problem because then the data collection process can be flawed. So hopefully the idea here is that you begin to think about the scientific method and how it's implemented in all of the research articles that you're going to read in this course, and start critically evaluating the entire process so that you get a better understanding of what the data that are being presented actually mean.